Bonjour tout le monde. Bienvenue à la 4e journée des conférences MS.
Hello, everyone, and welcome to the fourth day of the conferences of MS Connect. Today, we'll have as a guest Catherine Larochelle, who will be talking about any research on MS. My name is Louis Adam. I'm the General Manager of MS East and Atlantic Division. Thank you for taking the time to attend this important conference. As a start, I would like to thank all of our sponsors, Roche Canada, and contributing sponsors, Biogen Canada, Bristol Myers Squibb, EMD, Serono Canada, Novartis Pharmaceuticals Canada, Fendo Farm, and Sanofi Genzyme for providing the financial support to make this visual conference possible on MS. You can submit your question anytime during the conference, and you can also join the conversation online by using Twitter with hashtag MS Connect. Make your voice heard and respond to our daily poll on our Twitter page at MS Society Canada. This session is being recorded and will be shared at msconnect.ca. So let me introduce to you Dr. Larochelle, who is a clinician scientist at the Centre Hospitalier de l'Université de Montréal, CHUM, since 2013. Dr. Larochelle has been staff neurologist at the Centre de l'Université de Montréal, and since 2016, a clinical assistant professor and a researcher at the CHUM Research Center in the Neuroscience Department, and the research has been supported in part by the MS Society of Canada. To you, the floor.
I'm very happy to be here to talk to you about menu, metabolism, and microbiome in multiple sclerosis, news and research advances. These are my conflicts of interest, nothing major, nothing in line with the current presentation. And now for the goals of the presentation. We will do a review of recent data on the impact of data in MS, review of recent data on the impact of the microbiome in MS, and a review of recent data on the impact of metabolism in MS, so immune cells. This is a way of viewing multiple sclerosis. Here we have a human being with genes, normal genes, but in some cases, this person could be predisposed to multiple sclerosis. And there's an interaction between these genes and the environmental factors that are also factors that predispose someone. So the ones that we're familiar with are smoking, mononucleosis, and vitamin D deficiency. And we believe that there's an interaction between the environment and genes, and in some people in whom multiple sclerosis can develop, it can lead to an activation of immune cells, which in a radical way will attack the brain and the bone marrow and create lesions. Here you've got an image by magnetic resonance, zones where the signal causes an anomaly. If we look more closely at these lesions, what we see is that there is an infiltration in immune cells in the brain and in the bone marrow. Now, this is the most inflammatory portion of MS in particular. So what we call the relapsing remitting form of MS. And we get the impression that with age or with time, and in some people in whom it is more progressive, we have a shift in inflammation that finds itself more in terms of the brain and the bone marrow. And it feels like we're a little bit less efficacious in terms of treatment there. We're better in different phases. So what we're interested in today is how diet can influence all of these phases belonging to MS. In particular, we have recently, in the past few years, realized that among the environmental influencers having an impact on MS, there is the vitamin D deficiency, which is in line with diet. And then we have dysbiosis, in other words, the intestinal flora that's not exactly the same as someone without MS, and obesity, All of these are factors that are in line with diet. And we also have cardiovascular comorbidities, which means diseases such as diabetes, adult onset diabetes, high blood pressure, cholesterol. These are comorbidities, what we call comorbidities, associated with a less good evolution of MS. And it is in line with diet because it's something that we can modify through diet. So here I'm going to show you, just to convince you a little bit, that there is a link between obesity and MS. A number of studies over the past few years have shown this. Here we've got an example of a questionnaire sent to patients with MS and people who didn't have MS, and people needed to indicate approximately what their body looked like when they were 5, 10, 15, 20, 25 years old. And according to the study, people with a BMI, a body mass index that was higher up until 30, meant that they had a higher risk of getting MS. And clearly this form of this body or this BMI, it's not just a question of diet, but it's genetic as well. But there's some aspects that we're interested in. In other words, we can modify certain things through diet. What's even more interesting is that it's not even just connected with the arrival of MS, but it's also connected with the response to treatment. So a recent study showed that obese children have a less good response to treatment than non-obese children. First line medications don't work as well in obese children. And in the nodules, the studies tell us that children with higher BMI have higher retinal atrophy, and we believe that that reflects overall atrophy in the brain and bone marrow. And so children with a higher BMI, whether it be for dietary reasons or for genetic reasons or both, it means that the result is that there will be stronger progression in atrophy. So that's something that we can want to have an impact on. Now, if we look at cardiovascular comorbidities in MS, so cholesterol, diabetes, all these things are things that we feel that diet has an influence on. And these comorbidities are more frequent in people with MS. Apparently in the latest years, things are increasing. There's more high blood pressure with people who have MS than in people without MS. And in all of these comorbidities, they are associated with an increase in the risk of progression. And so it's associated with less good evolution, and it's also associated with more cerebral atrophy. That's what we're trying to avoid as much as possible. And we have more trouble in avoiding that with our current treatments for MS. And so you can see again that for obesity and other comorbidities, as I said, there are genetic factors that determine that. But the thing that we want to focus on is diet. So if we take a look at diet itself, which is having an impact on BMI and cholesterol, well, does diet have an impact on MS? And so there's a pediatric MS study. We like using children because they don't smoke, they don't take alcohol, and so it's a more uniform population. So what we see here is when we consume lots of saturated fats, what we call bad fats, so in fried foods and very fatty meats. And if we take more of that, we consume more of this, we have more chances of relapse in children with MS. However, if we're eating more fiber and vegetables, then there are fewer chances of having relapses in children. And that's interesting because it means that diet could potentially modify the risk of relapse. Here's another measure which says that in children not consuming enough iron, there may be more chances of developing MS. So is it simply a reflection of the quality of diet in general? The fact that that's the result? It's tough to say, but it's an interesting conclusion. Here we've got a very well done Canadian study. Almost 7,000 patients filled out a questionnaire on their diet, their food, and what researchers did is they divided people in quintiles. So they said, okay, are you in the top 20% of the people eating well, or are you in a lower percentage, the 20% of people eating less well? And then to decide whether people were eating well or not, they said, well, if you're eating a lot of fibers, it's better. If you're eating a lot of whole grains, it's better. If you're eating a lot of fruits and vegetables, that's better. And if you're not eating too many deli meats or meat, and in particular cold cuts, what we call processed meats, then what's the status of that? And the people ingesting less sugar. So all of those people with these criteria were considered to have better diet. And when we compared them to people with less good diets, what we realize is that there were fewer symptoms, MS symptoms, reported by patients in those with a better diet. There was less depression as well. And clearly it's difficult to say what's the chicken and what's the egg, because if somebody is depressed, perhaps they're not particularly interested in making healthy meals for themselves, but it is interesting to see a link between MS symptoms and diet. Now, in another study that was done, this is an international cohort, we have the same type of conclusions as what I was talking to you about with the pediatric and adult Canadian study. So essentially, food is better, diet is better, less fat, more fruits and vegetables, and the more that's the case, the less possible it is to get a whole lot of neurological MS-related symptoms and incapacity, neurologically speaking. And so, according to this study, if we have a better diet, we have a better body. So things are happening with the diet, but we find that the diet can have direct and indirect impacts on neurodegeneration. And that's where in research it's such interesting to try to understand what the mechanisms are, what in diet changes things. It's clear that's not one single thing in a diet that will have an influence on this. So if we take a look at the direct impacts, well, the food that we're eating and that we will turn into components, digest or break down, it means that there are relatively big impacts in what we can see on MS. There are also indirect impacts. That'll change the bacteria in the intestines, and it'll change their own production of components that could end up in our bloodstream and influence degeneration and inflammation. And for the impacts, the indirect impacts, like we said, a higher BMI, more cholesterol, high blood pressure, diabetes, that's not good. And so there's an indirect impact of diet that will have an impact on genetics, to say that, okay, at some point somebody will have a certain level of sugar and cholesterol in their blood, and that will have an impact on processes around MS. Now, if we look at basic science here, I think this is one of the most fascinating things that I've learned over the past few years, that I've realized over the past several years. In MS, immune cells are too activated, so they tend to proliferate and to want to produce inflammatory influence. But when an immune system activates, it needs a lot of nutrients, and it needs to change its metabolism. Whereas if it's pretty quiet, it doesn't ask much of its metabolism. But if we look at the T cells in particular, these are the immune cells that are very substantial to MS. If we look at a T cell, which is at rest, it doesn't need much. But an activated T cell, as we can see in MS or other autoimmune diseases, it'll completely reprogram its metabolism, and it will be very dependent on what's available in terms of nutrients. So glucose, amino acids, and they start to produce these inflammatory factors. So there's potentially a way of changing the profile of an immune cell by changing what we give it as nutrients and by changing its metabolism. Now, cancer studies are potentially much more advanced than our research in terms of the T cell metabolisms, because for cancer, it's the opposite of MS. In cancer, T cells are not activated enough, and the tumor is in competition, eats the oxygen and the sugar, and secretes something to tell the cells to quiet down. In MS, it seems to be the opposite, which means that the T cells are hyper-metabolic, and there will be a fairly abnormal metabolism compared to the health status. Now, what's even more fascinating is that all T cells, not all T cells, have the same metabolism. So in MS, we find that we end up with too many pro-inflammatory T cells and not enough anti-inflammatory T cells. In the pro-inflammatory, we have Th17, and then we have what we call the T regulators. And they are opposite in terms of their metabolism, which means that the Th17 depend on glycolysis and glucose, whereas the Treg are more favored when there's not very much glucose. And I find it rather fascinating to see that the T cells in our blood moving around and going into the organs, and according to the quantity of available glucose, they may or not become pro-inflammatory, which is fascinating. What's even more fascinating is that we can have an impact on this profile of cell. And there are people who have done studies in animal models, right? Because it's animal, not human. So in mice, and what they've done is they have put mice into what we call fasting mimicking diet cycles, which means that the quantity of calories were limited every so many days. So there were days when they ate normally, and on other days they didn't have calories almost all day. And so the mouse was sort of fasting on the days when there were not many calories. And then we looked at the animal model around MS, and we looked at how things happened in the mouse. Did they have neurological problems or not? And what we realized is that the mice with fasting cycles had fewer neurological problems than the others that did. And they went even further and they looked at the cells in organs, like in the spleen and the ganglia, and the mice subject to fasting, in those mice, their cells were too inflammatory, too activated, and then eliminated themselves compared to the regulator cells. And those cells tended to grow. And when you look at the brain, it means that there are fewer immune cells that infiltrated the brain to create lesions, and there was less damage to the cells in the brain, so less damage to the myelin, and the cells were happier and wanted to produce myelin more frequently. So clearly it's not something, this is, we're not going to tell people to start fasting, but it does give us a concept, which means that we can modify neuroinflammatory processes like in MS by activating diet. Now, there's a lot of research on this right now. We want to dissect the question around calories or components in food. What has a positive or a negative impact on inflammation? Here we've got some examples of suspects. Salt, for example. Salt is created or determined as pro-inflammatory. And so you also have the trans fats that could have an impact as well. So when you talk about bad cholesterol or bad fats, those are combined. And here you've got all sorts of bad fats. And opposite, on the contrary, we've got some good components, omega-3s, fatty acids that have an inflammatory impact. Of course, it can't just be any omega-3. We don't know if it needs to be consumed directly from a natural source or if we take it as a supplement that works as well. We don't know that yet, but it is clear that omega-3s seem to have an anti-inflammatory effect and seem to decrease the cells that are having an impact on MS. And then there are other components, flavonoids, for example. This is a word that we don't necessarily say very often. Well, I don't say it very often, the flavonoids. But if we look at flavonoids are components that we can find in a number of things like strawberries, spinach, blueberries, green tea, chocolate. And when we eat these components, when we consume these components, we can absorb them. They can find their way into our bodies. And then when they find their way into our bodies, we can look at a T cell very closely and we can see these components acting on what we call the AHR. It's an agonist. It's a molecule that is kind of like a flip switch and says, okay, we'll be pro-inflammatory or anti-inflammatory to a T cell. And so essentially, when we eat flavonoids such as these, they can get stuck to this switch and make the switch, the T cell, change so that it be more anti-inflammatory. And so again, that's fascinating. There are also what we call tryptophan metabolites that can be generated from broccoli and cauliflower type vegetables, but it can also be generated by intestinal bacteria. Again, tryptophan will have an anti-inflammatory impact. It does the same thing as the flavonoids. And so it has an impact on the cells, and it can even cross the blood-brain barrier, end up in the brain and in the bone marrow, and have a beneficial impact on the brain to say, okay, you don't have to get too affected. So this is produced by bacteria, intestinal bacteria. Now, this is a field of research exploding in a number of sectors, not just in MS, but in particular, clearly that's what's of interest to us, but it's the intestinal microbiome. What is the intestinal microbiome? Well, in the digestive system, we are in symbiosis with a number of bacteria. We've got a lot of bacteria, in particularly in the colon and in the small intestine. And these bacteria will help us do many things. They'll help us digest. They'll help us produce the metabolism that we need. And then we can coagulate. It helps with blood coagulation when we're injured. And it's the vitamin K in the intestine that is used. And there are many things that will influence the intestinal microbiome. Diet is not the only thing, but there are other things that will have an impact on the microbiome. Exercise, stress, antibiotics, age. There are many things that can have an influence on this. And we can have an additional influence through diet. Now, what's interesting is that diet and the microbiome, well, they talk and they are in line with the brain. What does that mean? Well, it means that the brain regulates the amount of food that we want to eat, what we want to eat, what we're going to consume, and will also send signals to the intestine with regards to digesting and not digesting. And the intestine produces many hormones that will communicate with the brain and that will give the brain some ideas and suggest certain things. So essentially, if we take a very simple example and we say that the Western diet here, for example, the North American diet, so to speak, fast food, maybe salty and sugary things, bad fats, well, that diet will decrease good bacteria and increase bad bacteria, the ones that we find that are bad when we have an inflammatory disease. It'll also change the quantity of components that we said that were anti-inflammatory and increase anxiety and depression, but it could also increase other processes, whereas a non-Western diet, for example, or a Mediterranean diet will have an impact on favoring good bacteria that will produce anti-inflammatory effects such as butyrate, and that will act on the level of the brain as well, and that should help against anxiety and depression and decrease inflammation. Here, let's look at this here. We're understanding things more and more. There's some things that we already knew that we didn't necessarily understand, so all of the mechanisms behind all of this, but I want to come back with vitamin D. For vitamin D, we know that it's associated with the risk of developing MS when we've got vitamin D levels that are too low at the point where we're taking vitamin D. Maybe our mother didn't have enough vitamin D, the low levels. There are all sorts of risks around vitamin D, but in addition to that, even at the time when we've got MS, here you've got some examples. If the levels of vitamin D are too low, then there will be more risks of converting to a progressive form. There will be more risk of more lesions and quicker atrophy. And so essentially there are studies done on vitamin D and multiple sclerosis, and they're very connected. And we know that vitamin D has an impact on neurological processes. But what's fascinating is that vitamin D also has an impact on intestinal microbiome. And so this is something that I learned very, very recently as well. Indeed, it's vitamin D. If vitamin D decreases, and this is the vitamin D, the level of vitamin D that we have in our body, but it also could be the quantity that we intake. And so it could play on several levels, but if we don't have enough vitamin D, then it seems to have an impact on the maintenance of the intestinal barrier. In other words, there's an intestinal barrier. We want the bacteria in our intestines, but we don't want the bacteria to infiltrate the blood. And so we've got a mucus level that protects us. And normally we sort of keep the bacteria in their place with antibodies. But when we have less vitamin D, it seems like that has an impact on this great barrier. And because of that, there are bacteria that manage to come in and stick themselves. They come in, they stimulate the immune system because they are sort of in the process of invading. I mean, we're not going to have a huge infection, but they are invading and getting very close and interacting too much with our intestine. And that will be associated with an increase in inflammation in blood, with inflammation signals that could be connected with inflammatory disease. Now, if we look at vitamin D, the microbiome, and MS, there are genes that are associated with the level of vitamin D associated with MS risk. So if we have some genes that mean that vitamin D levels are lower, then there will be more risk of developing MS. And these same genes are also associated with changes in the intestinal flora. So maybe if you look at these charts here, they may seem not substantial, but they're big for what is measured. So in people with vitamin D receptor polymorphism, the composition between some bacteria will not be the same in people who don't have this polymorphism. So polymorphism is a type of gene. And it's fascinating because it could make a connection between the two. Now, what proves that it's important to talk about dysbiosis and intestinal microbiome in MS? Well, amongst other things, there was a fascinating study that was done by a German group where they essentially took feces from patients with MS and their twins who also had MS or who didn't have MS. And then they transferred these feces to mice. These were mice that were at risk of developing MS, so predisposed, but weren't necessarily going to develop it. And they weren't going to develop it if we took out all the bacteria from the intestine. So all the intestinal bacteria was removed. They recolonized the intestines with the feces that had or didn't MS, and you see the neurological developments. They were much higher in mice with feces with MS than the non-MS group. And when they went to look at this information, they saw that some bacteria, the abundance was different in feces with people with MS. And in recent studies that were done, and you have to be careful here because these are very complicated. There are lots of things influencing the microbiome: diet, age, exercise, what they ate in the past 5 days, what they ate in the past year, what's the weight, etc. But there are some studies that were well done. And here, without delving into all details, if we look, the gray are people without MS. In red, it's all the people with MS who participated in the study, so 60 people, and you can see that there are a number of bacteria where the differences are huge between the gray and the red, so between people with MS and those without. And if we look at the people with MS, it's as if they recut the group in half. People who are untreated are orange, treated people are blue, and so it seems that MS treatment in some cases reestablishes the intestinal flora that resembles a intestinal flora in somebody who doesn't have MS, and you can see this for a number of different bacteria. In some cases, the treatment doesn't seem to reestablish intestinal flora, and there are other cases where the treatment does have an impact on flora. So we're far from having understood everything. We are far from being able to change everything and tell people what to do, but it's really interesting to see that there are changes in intestinal bacteria associated with MS and associated with the treatment of MS. Now, what's even better is that there are ways of modifying intestinal bacteria, intestinal flora. Because it's one thing to say, well, it won't go so well if your intestinal flora is not good, but it'd be great if we could work on the intestinal flora. And so there was a study done by Howard Weiner where they gave a probiotic to patients with MS, and it's very innovative. It's brand new. These aren't huge numbers, but it's extremely interesting. So when they gave the probiotic, they gave it to people who didn't have MS and to people who did have MS. two categories, two groups. And what you see is that when they are on the probiotic or not, red or blue, things change in the bacteria and in the controls and in the people with MS. And they also found that there was a bacteria associated with risks of having more inflammatory T cells. And this category was decreased in intestines of people with MS. And when we gave them the probiotic in question, and when the probiotic was stopped, then the flora retained or went back to what it was before. And so it's interesting because the realization is that we have to maintain flora. You don't just change flora once and then it's good for the rest of your life. And clearly people are working very hard to try to find a better way of understanding how we change the flora. Is there one antibiotic? Is there one thing? Is there something that's very specific that we can do? And that's where there's a lot of research. But essentially, the research is very promising to say that, yes, we can change intestinal flora. So I talked to you about all of that, and now I'm going to show you this slide again. The one that I showed you at the beginning of the presentation. I think I've convinced you that there are a lot of things associated with a diet, such as a lack of vitamin D, dysbiosis, obesity. All this has an impact on MS, and we also see that age has an impact on MS. And age, without wanting to delve into too many details, and there's chronological age, but there's also biological age. And so for chronological age, there's some people who are biologically younger than their age, and others are biologically older than their age. And you can look at not only the overall age, but you can look at the age of certain cells. So some people have more rapid aging in immune cells in people with MS than not. And so this can be associated with what we call inflammaging, so age-related inflammation. And there's also the fact that age is associated with the fact that there may be high blood pressure and diabetes because it's older. When we get diabetes and high blood pressure often when we are older. And there's also what we call change in methylation. And so what that means is that DNA will be more or less easy to read. And so some genes are more or less easy to express with age. It's what we call changes in methylation. When you combine all this together, we say, okay, with a diet, we've got maybe an impact, but how can we have an impact on age? It's very tough to have an impact on age. But yes, there is something potential that we can do on biological age. And again, because we're talking about diet, let's look at this. So what are the interventions that we call biological anti-aging interventions? Over many years, it has been known that when we limit calories, it'll increase life expectancy. This is a mechanism that's very felt, whether it be yeasts or whether it be in primates or in mammals. And here you can see mortality and control primates, so normal versus control. And then this is the normal diet. And in primates with a lesser calorie intake, they seem to live longer, so 35 years rather than 30 years in the lower calorie group. So potentially that could be the case for humans as well, even though it hasn't been tested. When we talk about calorie restrictions, obviously we're not talking about stopping eating altogether. We're just reducing a certain percentage of calorie intake. For example, instead of eating, if normally what you eat is 100%, then we'll just decrease to 90, 95, 85, 80. So it's not something that you should try to apply in yourself today, but it is something that is interesting. It exists, and it proves that there's a way of changing something when it comes to having an impact on biological aging. For calorie restrictions, that's not easy, and there could be side effects. In other words, if we don't eat enough of certain things, we will have a deficit and we'll be very sick. And so there are alternatives that have been explored. So is it really calorie restriction, or is it the fact that we eat less of one thing that has an impact on anti-aging? What's been found is that there is one amino acid called methionine, and methionine, if we decrease the quantity of methionine in a diet, we can then mimic the benefits of the calorie restriction by allowing animals to eat the number of calories that they want, but by limiting the amount of methionine that they might eat. It can't be at zero because it's an essential amino acid, so if we remove it entirely, there'll be other problems, but we can reduce it a lot in a diet, and we can reduce it by 40% or 30%. Here what you see are animal models with accelerated aging, and you can see the mice in which we decrease the levels of methionine. Their fur is nicer, they look healthier than in the mice where we didn't decrease methionine with the signs of aging. And here you've got another model where you see the back of this mouse, and they have this mouse has some back problems, versus the mouse which is a little bit better, fewer impacts associated with age. In particular, what was interesting here is that when we limit methionine, it's a way of also decreasing inflammation. So methionine restriction not only reduces inflammation, it seems to slow down aging, and it also has an anti-diabetic effect. So it decreases insulin resistance, and it has an effect to prevent diabetes, and it decreases cholesterol as well. So it's got an anti-cholesterol effect, which is rather interesting as well. Here, without going into huge details, I'd like to show you the methylation that can express a gene. Methylation as such that we do directly on the DNA and directly where there is a gene, and wherever there are signs to say we have to read the gene or not, then if we want to modify this expression, we have to add methyl groups, and methyl groups come from somewhere. What we call SAM, S-adenosylmethionine, and so it's a methyl group donor, and it becomes methionine expressed in a T cell, and that means that the T cell depends on the methionine around it, and methionine replaces it to change the gene expression. Now, we've been studying this for several years now, because when we reduce methionine in a diet, people and animals with cancer will have their cancer genes modified, and it slows down tumor growth. So there's going to be an anti-cancer effect, even if the effect is also anti-inflammatory, because inflammation is not good in cancer either. So all this together tells us that things are really interesting. So what we did is we asked ourselves questions and wondered if methionine restriction could improve neuroinflammatory processes in MS. And so here's what we did as a project with Dr. Russell Jones, a metabolism and T cell specialist who works in cancer and who is interested in anti-inflammatory diseases. What he showed here, and these charts are a little complex, but what he showed is that activated T cells need more methionine, and they will increase their metabolism to be activated, and the T cells that we consider to be bad for MS, so the Th17 consume a lot of methionine. Here we just decrease the amount of methionine in their cultures. We decrease their capacity to produce inflammatory factors such as the IL-17, one of the factors associated with Th17, and what we see here. And I also have a little trouble understanding what we're seeing in this chart here, but believe me when I tell you that what we're seeing here is that the gene has fewer methyl groups. And so by decreasing methionine in the cells, we decrease the quantity of methyl around the genes. And so we change gene expression. And we believe that because there is less IL-17 produced here, it's because the genes have signs to say, don't read this gene too much. So this is really interesting in cells, of course, but clearly what's interesting is to watch this in an animal model as well. So we've had the opportunity, Dr. Jones has asked us to test a methionine reduced diet in animal models for MS. And so in mice, where we had pro-inflammatory cells that will recognize myelin and attack the brain and the bone marrow. And what you see here are the amounts of neurological, or the quantity of neurological problems. And so the lower the better. And so mice around day 9, after the myelin peptide injection, they will begin to develop neurological problems. Whereas in the mice, where we simply decrease their methionine, they eat all the diet that they want, everything else is the same quantities of food, we gave them exactly the same things. Well, in those mice, just because we decreased methionine in their diets, we're talking about that we talk about stopping the disease for 5 days, and the disease is less severe than in the other mice. And when we look at the bone marrow and the brain of the mice, there are fewer pro-inflammatory cells that get to that point to cause damage. Now, not only does this work with diet, but it's also complicated in diet, like I was saying. So what's even more interesting is that if we block the enzyme changing methionine to the donor group, MAT2A, we get the same types of results. And this is really interesting because the MAT2A inhibitors were developed pharmacologically in some cases, and they are in trials in trials for cancer. And so if it's something that works for cancer and it could be good for inflammation, well, it could be something that could be transferred eventually to inflammatory diseases. So with everything that I showed you, what I'm showing you specifically is that research is ongoing. And the idea is not to say, okay, well, it's time to reduce methionine in the diets or to start to modify genes and that sort of thing. We can't say any of that because we're not at a point where we can actually understand what is important for humans and what is good for MS patients. But specifically, it is a start, and saying that simply the way in which we modify our food intake is a way of modifying intestinal flora, metabolism in cells, and based on that we can start to try to better understand what exactly is important and how we can target things, whether it be through pharmacological interventions or diet, and how can we then target the metabolism. Maybe this will bring to new therapies in MS that will then allow us to change things with regards to the risk factors, so obesity, diabetes, etc., where we can change comorbidities that are cardiovascular, and where we can have an impact on aging. And just in closing, something else that exists as well, when we look at metformin, maybe some of you are familiar with the glucophage. So this is an old medication that we use to treat adult onset diabetes, and it decreases the amounts of glucose or sugar in blood. And in people with MS, if we give the treatment, what we see is that before treatment, we had 3 groups. So control group, a metformin group, and a product reducing sugar group. And so we see that everybody has the same number of MS lesions accumulating over time, and then they have the same amount of acute lesions as well accumulating in time. So they look at 2 years before intervention, and then they get, some patients get the glucophage or the other product to decrease sugar, or then nothing, a placebo. And you can see that there's a huge lesion decrease in people who have received the medication decreasing glucose compared to people who did not get any glucose decrease or treatment. And there are fewer lesions. And so very much overall, diabetes treatment seems to improve lesions in MS. So that is also very promising. They are small groups. It's not to say that you should immediately start getting glucophage or something else, but basically, we can see the impact on metabolism and things that have an impact on intestinal flora through diet. And I think we're going to be able to see many new things that will start up in research. And to be even more convincing, well, when we look at the immune inflammatory cells that I've been talking to you about since the beginning, they are decreased in the same treatment, whereas with a metformin or glucophage, the cells increase. And so it is an impact, which is not just an impression. It's not just invented. It does indeed have an impact on immune system inflammatory impacts. And so in cancer, like I mentioned, cancer treatments are much more advanced, and they are talking about precision methyl and working on some diets. So we may get the impression that diet may be a little unclear and we're making general recommendations, but basically in some cases we can look at, in the case of methionine, in vegetables, some vegetables express very little, others a lot, same thing for meat, same thing for everything. And in the American diet, in I mean, in an American diet, there's a lot of methionine, and there are other diets that are lower in methionine, like the Japanese diet or the DASH diet for high blood pressure, the Mediterranean diet. And so if we want to do precision medicine or individual medicine, we can make recommendations to people based on what they like to eat, based on their diet, without changing absolutely everything, but being very specific in certain things so that people can then reach the targets that we are expecting to reach. So we're not there yet, but it's just to say that this is definitely something that's really interesting and quite promising. And so with that, I'd like to thank all my collaborators, in particular Dr. Russell Jones, who initiated the project that we're doing on methionine in the animal MS model. Nathalie Arbour and Alexandre Prat are my collaborators and esteemed colleagues. Dr. Vertype is my supervisor. Dr. Jack Untel is a collaborator, has helped students constantly, young researchers in MS, Dr. Duquette and Dr. Girard, also at the MS clinic for the CHUM, and they constantly help in research. They are constantly helping us move forward, and they are encouraging us, and they do their own research, obviously. And this is my research team at the CHUM, my students and lab assistants. So young, brilliant people who are working hard to find more things and more solutions in MS to better understand it. So thank you very much for your attention, and I'd be happy to take your questions.
Thank you, Dr. Larochelle. It was extremely interesting as a conference. I didn't know the term Western diet. It's very interesting. So I will now refer to this term, actually. Thank you. I have a first question. What do you think about the keto or paleo diets for MS patients? Whereas we don't have any data concerning those keto or paleo diets. We don't know whether this is a nuisance for MS, but you need to be followed in nutrition if there are some dietary lack of essential nutrients, for example. We have to make sure that there's nothing showing lower nutriment. So we have to make sure that everything is lower than Western diets. Often there is a lack of nutriment, so we refer to the Mediterranean diet. We increase vegetables, fish, and so on in order not to create any lacks. Thank you. We're waiting some questions. That was a question that came in. Is it normal that I don't see any other question? So here's another question. When we're taking vitamin D supplements for a deficiency in vitamin D, is it preferable to take them all together or different over the day? At different times in the day, well, there's no difference in regard to the time during the day. We prescribe it at once a week, for example, larger doses, 10,000 units once a week versus a daily dosage like 2,000 or 3,000 units daily. Currently, we don't have any evidence that one diet is better than another. What we're trying to do is to get into a minimum level of vitamin D so that there's no deficiency or insufficiency in vitamin D. So it is all right to take it once a day or even once a week. Thank you. I'm just waiting for the next question. We're going to give ourselves a few minutes just to see if other questions are going to come in. It's not so easy with pre-recorded events, for example. Yes, indeed. There's a new question. What food actually contains a high level of methionine, and how can we reduce it in our diet? Well, once again, this is something think that if we were to apply it, we would potentially have to look at this with nutritionists because we can't say that the vegan, Japanese, or vegash actually contain less methionine than the Western diet. Generally, we would say that what has most methionine is red meat, and it depends on a part that we're using it, and also fruits and vegetables. Brazilian nuts, for example, also contain an enormous amount of methionine, and vegan diets actually use those. So it's a variable thing. But when we tend towards a vegan or Japanese or DASH diet, we're going to have less methionine. Thank you. So I'm still waiting for the next question. What food is most pro-inflammatory or anti-inflammatory for T cells? Well, this is a very good question. There's not necessarily one type of food, but components like trans grass food, for example, a bad fat. So this will increase the activation of T cells. There was a study that was carried out by PhD students, and they had them feed themselves on fast food for a week, for example, and then in their blood cells they saw that T cells were more inflammatory after 5 days. So it's not necessarily like one particular food, and with red meats, for example, in Australia, their studies showed that whether it was grass-fed or grain-fed, bovine meat could even have a beneficial effect when it was low on saturated fat. So we're talking about trans fat, saturated fat, and so on. Anything that's high on fat or high in salt, for example, this will push the immune system in the direction of pro-inflammatory. It's not because you eat something sweet one day that it is going to trigger that, but we're talking about chronic inflammation, and it is a main character that is created by the Western diet. Thank you. Is there a test for intestinal flora or the microbiome? Yes, well, not yet. We could take the feces of people. There are machines that are able to sequence all of the bacteria that are in the feces samples as part of the DNA that is different from another. We can take a look at the different bacteria. We can class the groups by species and so on, and identify which bacteria we're talking about. Yes, we can take a look at the intestinal flora of someone, but the people who are specialists, say that it is a difference when we sequence some flora, with the opposition of a biopsy of the colon. So we have it at the duodenum, for example, and more in the colon area. Yes, so we can measure it using a sample of feces. It doesn't represent 100% of what the presence is. If a person, for example, is taking antibiotics and a few days later, it may not be the same thing that is observed observed according to what we eat also. But in a few years, we can have some clinics on, regarding microbiomes, and they with recommendations, makes such and such a change in your diet, and then will sequence the bacteria of your feces, and we'll see what goes on. It's not necessarily ready, but this is part of the further examinations. Another question: when do you estimate that the recommendation could get to medicine with respect of nutrition? Well, this is very difficult, because when you're talking to nutritionists and what they tell us is that it's very difficult to conduct studies on diets on human beings. We love eating, we have pleasure eating, it's difficult to see what a normal portion is, and so on. So we could use what people have scanned or written on what they've eaten, and have an intervention and apply it at all times. It can't always happen that way, because when you go to a restaurant, outside of a COVID period, it's difficult to figure out what is given to you at times, and you don't have control over it. And the diet, well, it's not always the same at all times, because we want different things and we eat different things. So it's difficult to figure that out and conduct studies on that. I would tell you that my most obvious recommendations would tell you to avoid fast food, salt, and so on, with respect to all of the different aspects of your health. You can make some recommendation, but when you have a person sitting in front of you and who is an MS patient, it will be difficult and will take a few years before we go ahead. So I have one last question for you, another excellent question. If you choose a diet, what is the duration of the effect, and how long does it take to see any difference? Well, it is a good question indeed. It all depends on what you want to measure. It depends upon the diet, and it depends, well, for example, if you take a look at what you eat, and this will have an influence on the levels of sugars and amino acids, according to what I eat. So the immediate effect will be faster. And other cases, such as inflammatory parameters, for example, 5 days of eating fast food was sufficient to see the inflammatory profile of the T cells. But if we want to say that we have an effect in order to reduce any inflammatory effect, MS is something that stays on for a long period of time, and people that follow the recommendation and abide by them during the entire duration, or the entire number of years during which the illness is still active. They've made an intervention like fasting intermittently, for example, and avoid some food during 6 months, and there was some impact on the inflammatory effects. But it's difficult to follow. It's difficult to go through the day without eating at all, or with very low calories. With probiotics, in 2 weeks, for example, you have some changes. This was like adding lactobacillus extract and other probiotics for 2 weeks. But once you stop, 2 weeks later, the samples gave to some results before using the probiotics. So there's no long-term effect. So thank you very much, Dr. Larochelle. It was extremely interesting. You're always very interesting to listen to. It is a topic that is fascinating. There is a discussion group that will follow. If you cannot join this session, it will be a session tomorrow by Dr. Marcia Finlayson on MS fatigue management, the state of the science. I would like to indicate a contributing guest sponsors, Bristol Myers Squibb, whose new patient support program called the B program will be launching soon to support people. Now you go on Zoom, you will see it at the bottom of the screen. You'll have the screen to participate to the Zoom session in about 10 minutes. So thank you to all of you for participating to this very interesting conferences. Have a good rest of the day and good conference day tomorrow. Thank you.